mTOT stands for mitochondrial targeted thiazolidinediones. The thiazolidinediones were the first insulin sensitizers that were discovered empirically about 30 years ago. Nobody knew how they really worked. And what we've done is we've used a photoaffinity probe and some unique approaches to identify where these compounds are actually binding in the mitochondria. From that, we identified a complex in the internal mitochondrial membrane that has been unappreciated to date. This is a complex of proteins, some of which have been disclosed, some of which we haven't talked about, but which are in the internal mitochondrial membrane or an important control point at the crossroads of metabolism. And let me just show you what I'm talking about. In each cell, you have these organelles that are called mitochondria. And they are, many of them in different cells, composed of a series of membranes. An outer membrane, an inner membrane, and then space between the membranes on the inside and out. And in between these membranes is all the important control of metabolism, separated by those membranes in mitochondria in every cell in your body. And what the mitochondria are doing, if we place the mitochondria now in the context of the complete cell, and we'll just for this purpose draw it as if there's just one mitochondria in this cell, is they are controlling the control center, which is in the nucleus, by the signals that they provide metabolically. And most cells will use, for example, the glucose, glucose that's in the circulation will go into the cells through various, depends on the cell how it gets in. But this 6-carbon sugar is broken up into 3-carbon pieces that then have to go into the mitochondria. mTOT, where the insulin sensitizers are binding, is part of a complex that's in this internal membrane. And I'll just draw it here. It's actually a collection of a number of proteins. We haven't disclosed all of them. There are 2 particular ones that we and others have studied that seem to form an important component of what's called the pyruvate transporter. Pyruvate is the breakdown of this 6-carbon sugar into a 3-carbon source that then has to go across, it diffuses through this outer membrane, then it has to get through this inner membrane in order to enter into metabolism. And the first thing that happens when it enters here is it's converted into a molecule that's called acetyl-CoA. And we can measure that. We can measure the effects of drugs on that. And this is at the crossroads of all metabolism because this little piece here is what is involved in amino acid metabolism that gives rise to proteins, as well as fatty acid metabolism that gives rise to lipids, and of course these carbohydrates. And this can also, through a series of reactions inside the mitochondria, be oxidized to carbon dioxide, which can be released. Now the important thing here is that this metabolism is very tightly controlled. If it gets out of balance, if there is too much flow of carbon into this oxidative machinery here, then there is not the balance between this tightly controlled cycle and what's called the electron transport chain that generates energy that runs all cells. This is at the basis of the metabolic dysfunction that occurs with too much caloric intake and with aging, and as the basis of all these aging diseases. As a function of this, the cell will reduce its response to insulin, it will reduce its ability to metabolize, it will give rise to weight gain, dysfunctional adipose, and various tissues. This will give rise to the loss of cell function and the nerves where there's less synapses, there's less functional cells, less ability to clear out destroyed and old pieces of protein. And so you can see very readily how this is at the basis of what the problem is with these diseases of aging. mTOT is how the insulin sensitizers stop this entire reaction by bringing back into balance the level of metabolism, removing it from that situation where you have too rapid flow of carbon here to one that can be more manageable without causing these detrimental effects that are causing the diseases that we want to prevent.